everyone, thank you so much for clicking onto the video. Here are today's learning objectives. So we're going to look at how amino acids have both acidic and basic properties and the formation of Zwitterions. So by the end we should be able to draw the structures of amino acids as Zwitterions and the ions formed from amino acids in acid solution and in alkaline solution. So now we're going to talk about amino acids. 50 or more amino acids join together to form long chain polymers called proteins, and these are vital for living organisms. They have two functional groups, and one is a carboxylic acid and the other is a primary amine. And there are 20 naturally occurring amino acids. They're described as either alpha amino acids or 2 amino acids, which means that the amine and carboxylic acid groups are attached to the same carbon. And all amino acids are optically active except for glycine. Now, the general formula of an amino acid is the amine group, and then we have a carbon, an R group, a hydrogen, and then we have the carboxylic acid. And R can be anything, it can vary. So for example, R is equal to a hydrogen for glycine, or R is equal to a methyl group for alanine. So now we can see why glycine won't have optical isomers, because if we draw out the structure of glycine, H2N, and then carbon with 2 hydrogens and a C double bond OOH. And so this carbon here, which is normally the chiral center, if we were looking at other amino acids, it's not a chiral center here because we have 2 hydrogens, so we have 2 of the same group on this carbon, and so we don't have 4 different substituents, and so we don't get optical isomerism. And so for an example of a 2 amino acid, we could have 2 amino ethanoic acids. So what that would look like would be if we had an amine, and then a carbon, and then we had a hydrogen and another hydrogen, and then C double bond OOH. So that's how we can get ethanoic acid, because we've got 2 carbons, and amino because we've got the amine. And this is actually our glycine, but the systematic, the IUPAC name is 2 amino ethanoic acid. And so one thing you'll notice is that the carboxylic acid takes priority. So it becomes the root of the name. And also that means that the carbon which is attached to the double bond O and OH, this carbon is labeled number 1, and so this is 2. And so this is how we get 2 amino ethanoic acid. So we're treating this amine group here as the substituent. Now we're going to introduce Zwitterions. So amino acids have both an acidic and a basic functional group. And this is the key thing about amino acids. So the carboxylic acid group of an amino acid will tend to lose a proton and act as a Bronsted-Lowry acid. So what we have is the COOH group, which is a carboxylic acid group, and this will be in equilibrium with the COO minus ion and also the H plus ion, and so it loses the proton to act as an acid. However, the amine group will tend to accept a proton and act as a Bronsted-Lowry base. So what we have is the amine group reacting with a H plus, so it accepts a proton, and this is in equilibrium with the ion NH3 plus. So it's really important to remember which group accepts a proton and which group loses a proton. So we have the carboxylic acid group losing the proton, and so it acts as a Bronsted-Lowry acid. And this is quite easy to remember because a carboxylic acid we associate with acids. And the amine group, on the other hand, will accept the proton, and so it acts as a Bronsted-Lowry base because That's the definition of a Bronsted-Lowry base. And we say that amino acids actually exist as Zwitterions. And the definition of a Zwitterion is an ion that has a permanent positive and negative charge, but overall it'll have a neutral charge. So basically the positive and the negative will cancel out. So for example, if we were going to draw the Zwitterion of serine, we have serine here, and you can find this in the data booklet. What we do is we add a proton to the amine groups and take away a proton from the carboxylic acid groups. So what we get as the Zwitterion is an NH3 here, and so this would now be positively charged because we've added a proton. And then you'd have the CH as usual, and then here you'd have COO minus because we would take off this H plus of the carboxylic acid group, and then we'd have CH2 and then OH. Now it's really tempting to take off this hydrogen here, but we need to remember that this alcohol doesn't do anything. It's only the carboxylic acid group that acts as an acid, a Bronsted-Lowry acid, and gives away its proton, and so we only get CO minus ions with the carboxylic acid group and not the alcohol, so it remains as OH. So this would be our Zwitterion. So you can see that it has a positive charge here and a negative charge here, but overall, because we've got one positive and one negative, they balance out and we have a neutral charge. And we're now going to quickly just look at the general formula for a Zwitterion. So we know that the general formula of an amino acid looks like this. So we have NH2, so this is our amine group, and then the carbon with an R group, which can vary depending on the amino acid, and then we have the carboxylic acid group. So this is our amino acid, very generally, and then our Zwitterion would look like this. So we'd have NH3, and this would now be a positive ion, and then we'd have the carbon, the R group, and then hydrogen, and then we'd have C double bond O, and then O minus. And this is our Zwitterion. So this is in general. So we can see that for serine, the R group is actually this CH2OH here. Now we're going to look at amino acids in acidic and alkaline conditions. So we've just discussed how amino acids can exist as what we call Zwitterions, so ions that are that have a positive charge and a negative charge, which balance out to make the whole compound and the whole ion neutral. And so when we place them in strongly acidic or alkaline solutions, we can form protonated or deprotonated ions. So first we're going to look at strongly acidic solutions. When we put amino acids into strongly acidic solutions, the lone pair of electrons on the nitrogen of this amine group accepts a proton from the solution to form a positive ion. And so we say that the amine group has been protonated, and so the whole amino acid has been protonated. So what we have is that originally we have an amino acid that looks like this. So we have NH2, and then a carbon, an R group depending on what amino acid it is, and a hydrogen, and also a carbon double bond O and OH. So this is our amino acid. And so now what we have when we add it into a strongly acidic solution is that this nitrogen then accepts another proton. So we have NH3, and this will also be a positive ion because it's accepted that proton, and the rest of it is all the same. So we still have this carbon, and then we still have this carboxylic acid group. So this is our protonated ion. However, when we put amino acids in strongly alkaline solutions, the OH group of the carboxylic acid loses a proton to form a negative ion. and we say that the carboxylic acid group has been deprotonated. So again, we have the original amino acid. So we have NH2, and then the carbon, the R group, and the hydrogen. And then we have the carboxylic acid group. So again, this is our amino acid. And now we're going to form the deprotonated ion. So we still have this NH2 and this carbon with the R group and a hydrogen. But now this carboxylic acid has turned into a COO minus because we've lost that proton. And so this is our deprotonated ion. And why this makes sense is that for the strongly acidic solutions, if we have a really acidic solution, what that means is that we have lots of H plus ions in the solution because we know that pH is equal to minus log to the base 10 of the concentration of H plus ions. And so strongly acidic solutions have a very low pH, and that means they have a very large concentration of these H plus ions. And so if we put an amino acid into it, what the nitrogen will want to do will be to accept protons. We'll accept a proton because there's so many protons floating about in the solution. And nothing will happen to the carboxylic acid group because we can't take any H pluses away because nothing's attracting a H plus. There's loads of H pluses in the solution. And when we look at the strongly alkaline solutions, what we have is that we have OH minus ions. And these OH minus ions will then not do anything to the nitrogen, so the amine group, but with the carboxylic acid group, they'll be attracted to H plus ions because it wants to be neutral. So it takes away that H plus ion from the carboxylic acid group. So we get a deprotonated ion. So we're just going to look at a quick example, which is to draw the species formed by aspartic acid at a low pH, and it has this structure. So at a low pH, that means we have a very high concentration of H plus ions. And so what this means is that it's an acidic solution. And because there's so many H pluses floating about in the solution, that means that we add a H plus to the nitrogen of the amine group. So the species we form is a protonated ion, and we form NH3 plus because we've added a proton, and then CH, CH2, COOH, and then COOH. So nothing happens to the carboxylic acid groups, only to the amine group. And so we form a protonated ion. Protonated just means that we've added a proton.